But today we are going to talk about details about the way that we generate antibody diversity. And I left you off with this problem on Monday. The problem is that it is estimated that we can make a large number of antibodies. Here I tell you 10 to the 16th. On a couple of other slides, I'm going to give you slightly different numbers. The moral of the story, whatever the number is it's a big number. Yet we have a much smaller number of genes, and so there's this question of how we can get to this huge diversity of antibodies with such a small number of genes. I'm going to talk through a lot of this sort of as theory today, and as I said, on Monday we're going to get into a lot more of the nitty-gritty details. And for me, in thinking about this, one of the reasons why I can understand the details and I can focus on the details is because I know why they're important. And in understanding why they're important, it's also good to think about some of the theories people have come up with for this problem. Because in fact, since about 1900, people started to realize there was probably a problem here, numbers-wise. They calculated different numbers at different times, but they realized something was up with this amazing diversity of antibodies. And they came up with some theories. And I want to talk through what their theories were and why we learned now that they are wrong, so you can understand what are the important pieces of a theory that's right. What makes the other ones not right. When they were trying to come up with a theory to explain this process, they had a few other observations that they had made. One of those observations had to do with this idea of the primary and secondary response. The idea that the second time that you saw a particular antigen, that the response improved. And so when past scientists were trying to come up with ways of making diverse antibodies, some of their theories, they tried to include ways that they could get improvement over time in their theory. They also knew that class switch occurred, and they knew that affinity maturation occurred. And so again, in some of the theories that I'm going to show you that ended up being incorrect, some of the weirdness came because they were trying to come up with mechanisms for class switch and for affinity maturation. I'm going to show you two theories that were wrong, and we're going to talk through kind of why they're wrong and why someone might have thought of them. And you might worry that I am kind of making fun of these past scientists. Both of the past scientists who I'm going to talk about, actually all three of the past scientists I'm going to talk about, won Nobel Prizes for something else and are pretty famous for other stuff. So they don't need anybody sticking up for them. They're in good shape as is. And the first person who really came up with a theory to figure out how exactly antibody diversity occurred was a guy named Paul Ehrlich. And he came up with this theory in about 1900. Again, just so you know, yes, this theory did prove to be wrong. However, Paul Ehrlich did win the Nobel Prize for his work on immunology. Remember the stain that we looked at in lab 1 that let us distinguish the different cell types in the blood? He discovered that stain. He also discovered the first antibiotic and basically started antibiotic therapies. So it's okay that he was wrong. And so Ehrlich had this idea that perhaps we would have a cell, which you can see in the upper right here, that might bind to some antigen, and those little fish or octopus-looking shape things are the antigens in this case. And so we might have a cell, it binds to antigen. When binding occurs, as you can see with this little black fishy thing, then the cell makes more copies. And eventually those little copies, and you can see again more binding, results in more copies. And eventually those little things fall off, as you can see in number 4. And those falling off things are the antibodies. And so he came up with this because he was like, okay, this is a way that we can get improvement over time with our response. This is a way we could get better binding over time. And so this is what he was coming up with. We now know that it's not just one cell with a bunch of different kind of side chains that it makes more of. This is not the way it's going to work. Some other scientists came up with another theory a little bit later. This was most famously promulgated by Linus Pauling. You may have learned about Linus Pauling in chem. Linus Pauling won the Nobel Prize for, and I'm quoting the Nobel Committee, discovering the nature of the chemical bond. So he's a pretty big deal in chemistry. He actually is also the person who first showed that a human disease can happen because of an abnormal protein with sickle cell. And he's one of the few people who's won 2 Nobel Prizes because he also won a second one for work with nuclear disarmament and sort of activism. And in fact, this isn't even his worst mistake. He made a structure of DNA and was competing with Watson and Crick, and that DNA structure was bad. This isn't even his worst thing. So I'm definitely not making, Linus Pauling can take it if I'm making fun of this theory. So Linus Pauling's idea of what happened for antibodies is that there was some antigen, which is shown here as a black circle, sort of with a bomb shape to it. And that there was some protein that was an antibody that was unfolded, and that antigen served as a template to allow more and more and more folding of that protein. And so the more antigen you had around, the more sort of training your protein would get to make this antigen and to let it go forward. So as you look at this theory, and as well as the theory above that I showed you before, what kind of problems do you see with them? There are a few we can come up with. For those of you who are taking biochem, how does this go along with things you've learned about how protein folding works? Yeah, Emilio.
What interactions are going on? What's binding to what? How is it binding to stuff?
Yeah, so there are some questions about sort of general biochemical interactions here, how things are binding together. What about just big picture biochem? Yeah, Jordan.
Protein structures determined more by like the primary sequence than what it's doing when it binds to something.
Yeah, protein structure is defined by the sequence of the protein. You don't have a protein that just sits around unfolded and that gets folded around stuff. This is the opposite of how biochemistry works. And so we know that now, but in 1940, Pauling didn't have that figured out. So we can't actually have our protein folding around this. We, in fact, have to have a protein in advance that already is able to bind to our antigen of interest. There's one other big problem with both of these theories. Works a little bit better with Ehrlich's, or it's a little easier to see with Ehrlich's, but it actually is a problem with both of them. So can you think of any other problems that you see here? So if we think about this cell, it's a really, really specialized cell at this point. After all of these 4 steps, that cell has really specialized itself. It is like our amazing super cell that can bind to this particular antigen. It's a really special cell for us, right? At this point, after all these steps. What happens if that cell divides? Is it going to transmit this antibody to its daughter cells? Not exactly. I mean, maybe you can imagine it sort of moving, but probably not. Yeah, what were you going to say, Malik?
Oh, no, I was just going to say that it wasn't going to vertically.
Yeah, it's not necessarily going to transmit this to the next generation of cells. And if this cell dies, then it's like we lose our entire response. In fact, you guys, when you learned sort of some basic evolution in intro bio, probably learned about this guy named Lamarck. Who's was Lamarck, and what was Lamarck's idea? Yeah.
Essentially, a giraffe could grow a long neck just by stretching itself, and then its progeny would inherit this physical trait because it was a physical trait.
Yeah, so Lamarck had this whole idea of inheritance of acquired characteristics. You would have a trait that you needed, and so you'd have it, maybe because you were a giraffe and you got a long neck. And then your progeny would just have it because it was useful. And we sort of make a knock on Lamarck now. And we say that, we all know that Lamarckian evolution is not true, and why is Lamarckian, what's sort of a problem with Lamarckian evolution? Yeah, Molly? Yeah, how the heck do you transmit a long neck from one progeny to another? The only way you can transmit things from one generation to another is through DNA. This is actually just inheritance of acquired characteristics on a cell level. And so just like with Lamarck, the thing that's the real problem with all of these strategies is that they don't address anything happening with DNA. And we can't see a way for there to be progeny cells, further generations of cells, that are going to be able to carry on the production of this awesome specialized antibody. And so what that should tell us is that the mechanisms that we are going to learn that are going to be important for diversity of antibodies are going to have to go to the DNA level. If we want them to be able to transmit to future generations of cells, then we need them to be happening with DNA. And in fact, I've already shown you the clonal selection theory, and I will show you the clonal selection theory again a little bit later today. But the basic idea of the clonal selection theory is that we have these stem cells in our, that are making up our immune system. They're the hematopoietic stem cells coming from bone marrow, and they do this process of diversification, of making lots of different antibodies, and it's a process that involves messing with DNA. And then later on, if they become exposed to antigen, then those cells will expand. So the first part of this is making this very diverse set of clones. I'm going to repeat this again because this is an incredibly important piece of information in immunology. And I guarantee you that if you ask your non-science friends, this will be one of the most common misconceptions they have, and it's a thing that might trip you up on exams because you probably have this misconception, and I'm going to try to get it out of your head, but you might fall back on it, so I push it a lot. This diversification process happens before you get infected. It happens before you're born, throughout your life. You're doing it right now. You're making more diverse cells that are going to make your antibodies. And so you can imagine that you basically have one of every possible kind of antibody-producing cell right now. For a long time, people were worried about it being zero. It was actually because we didn't know how to measure rare cells. Now that we figured out how to measure rare cells, people can actually go in and do the measurement. And it turns out it's not even 1. It's like between 5 and 10. But we'll just say 1 for right now. You have one of every possible antibody-producing cell right now. You have an Ebola antibody-producing cell right now. You usually only have one of them. So if you got Ebola, it couldn't make very much antibody. You'd have to wait for it to expand later. You have one antibody-producing cell against Zika right now. You have one antibody-producing cell against the flu that hasn't even happened that's going to happen in 20 years. You have one of every single one right now. It's not enough to make very much antibody, but if you get infected, that cell is going to expand. So this diversification process that we're going to happen today is happening before infection occurs. So we need to think a little bit about how we solve this antibody diversity problem. And so again, the problem is about how we're going to make 10 to the 16th different proteins when our genome only has 2 times 10 to the 4th genes. This slide is in here for your later studying. It's a nice outline of the answer to how we do this. You can see that there are two answers something called combinatorial diversity, and something called junctional diversity. And under each of those, there are two different pieces. We're going to walk through all those two pieces today. So obviously you don't know what all these words mean right now. But when you come back to study for the exam, this is going to be a very helpful outline slide for you. And so the first thing that we really need to talk about, the first way that we address this problem is through the process of combinatorial diversity. And it turns out you actually already kind of know about combinatorial diversity, though you don't realize it. And one of the things that I really liked from my undergrad immunology class is that my professor actually used a metaphor to explain combinatorial diversity, and it like made it click. I remember I'd read the book and I was like, why are they teaching me this Greek? And then suddenly when he did the metaphor, I was like, oh, that's all it is. So I'm going to give you a similar metaphor. So let's imagine that I decide this whole science thing isn't going to work out for me, and that instead I am going to open a restaurant. Sadly, I do not know how to cook very well. This is not an overstatement. And so let's imagine that I know how to cook only 3 dishes. Let's imagine that I know how to cook chicken, beef, and broccoli. I know how to cook 3 dishes. And in fact, my restaurant can have 2 things on the menu. My restaurant can have chicken and broccoli, or beef and broccoli. That's it. That's all my restaurant has. Now let's imagine I learn to cook 2 more dishes. Let's imagine that I learn to cook pork and that I learn to cook carrots. I've only learned 2 new recipes, but I'm going to count my restaurant's meals based on combinations of those things, not on individuals. So even though I've only learned 2 new recipes, now I get pork and broccoli, and I get chicken and carrots, and I get beef and carrots, and I get pork and carrots. So even though I only learned 2 new recipes, my restaurant got 4 new dishes because I'm counting the dishes as combinations of recipes and not as individual ones. If I again start to learn some more dishes, you can imagine that I start adding just 1 additional recipe and multiplying it out times all the combinations I can do, and suddenly I can have a very diverse restaurant with a pretty small number of recipes because I count combinations as being the dish, not just the individual recipes. Does that make sense to you? That's combinatorial diversity. And in fact, you've already kind of seen one level of combinatorial diversity for antibodies, which is that antibodies contain 2 copies of an identical heavy chain and 2 copies of an identical light chain. So in fact, you have 2 genes, the heavy chain gene and the light chain gene, and you can pair different versions of them to get different antibodies. So just like we have chicken and broccoli, this could be heavy chain number 1 and light chain number 1, and heavy chain number 2 and light chain number 1, and heavy chain number 3 and light chain number 1, and heavy chain 1 and light chain 2, and heavy chain 2 and light chain 2, and heavy chain 3 and light chain 3. So just by combining different heavy chains and light chains together, and with each different combination giving us a different antibody with a different specificity, we can get a much bigger number of antibodies with a somewhat smaller number of genes. Is this making sense? So that's kind of the first level of combinatorial diversity. But then some scientists made some further observations about antibodies. They found that they could find antibodies that have the same constant region with different variable regions, which you see here on the left with the different shades of blue. You can also find the same variable region with different constant regions. And those are our 5 MDs, give everyone apples, constant regions. And so they hypothesized, what if the DNA that makes up the variable region and the DNA that makes up the constant region are separate pieces of DNA that get combined in different ways? And so we have these small gene pieces, or mini genes they were called, that get combined together to make the big genes. And so instead of having to have a whole bunch of different antibody genes, we have mini genes that we just combine in different ways to give us things. And so the hypothesis that came out of this was the Minigene Hypothesis, where they said, okay, maybe you have some variable region genes that pair with different constant region genes. And maybe even if we take this a step further, you have multiple different pieces of your variable region genes that get put together with the constant region. So you can use just little fragments of DNA in different combinations in order to make your variable receptors. And in fact, that is the case. We do know now that that is how this works. So the way that this will work is that we are going to have segments. You'll see these segments a little bit more as we go on, that are known as the V, D, and J segments, as well as the C, or constant region segment. And during development, each of our B cells will take a different V and a different J, or a different V, a D, and a J, and combine them together to make different receptors. And so here you can see we have one cell that made V1 and J, that used V1 and J1 and made this receptor. One used V1 and J2, one used V2 and J1, one used V2 and J2. So we have basically every possible combination of our V's, our J's, and in some cases our D's, in order to make these receptors. And in fact, the idea was, spoiler alert, was shown to be true, that the cell was actually going to choose, I say choose with air quotes because the cell doesn't have a brain to make a choice, but the cell is going to basically pick one V, one J, join them together so that they are touching. You can see that there's nothing in between them now, they're touching, and actually cut out the DNA in the middle and throw it away. And the idea is that there are a lot of V's, a lot of D's, and a lot of J's. And it's a numbers game. Every cell is randomly picking a V, a D, and a J. And there are just so many cells that every combination ends up getting hit at some point in time because there are so many cells. This is where people's not sitting. Are you sure that it's random? Yup, it's totally random. This is a random selection of V's and J's, or V's, D's, and J's that are combining together, and it's just a numbers game that we get them all made. Yes, Molly?
Is the inclusion of D segments less frequent? And if so, why?
I'm going to talk about this blah, 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 blah. I am going to answer that specifically as we go on. So you will see the specifics of what's up with the D segment. Yep, Jordan. When they were described, they stood for variable, diversity, and joining, although that's not particularly important now. No one really thinks about it with those terms. But they did originally stand for variable, diversity, and joining. Yep, Malik.
And is it usually like two things that combine, like just V and D or V and J or D and J?
We'll get to that in a little bit too. Great questions. So before we answer those questions, I want to tell you a little bit about how we proved this hypothesis to be true. We didn't. This was before I was born. But, you know, it's how you talk. So there was a scientist named Tonegawa. And Tonegawa There's a lot of things about him that are really interesting. But Tonegawa did the experiment that eventually proved the Minigene hypothesis. So if the Minigene hypothesis was correct, you could imagine that you have a cell with normal DNA. Maybe this is a cell that has not yet undergone this process, or maybe this is a not immune cell, so it shouldn't be making diversity processes, doing these antibody diversity processes. So in different versions of this experiment, in different parts of this experiment, sometimes people will use things like liver cells as non-immune cells. Sometimes you'll see it done with sperm cells as cells where you would expect the DNA should not be messed with. You can see a bunch of different cell types here. So basically the thing that's important here is that it's sort of some non-immune cell. In those cells, you should have, minigene number 1 and minigene number 2 far apart in the DNA. And if you were to take an antibody producing cell, that antibody producing cell would shockingly produce antibodies. Those antibodies would be encoded by an mRNA. And in theory, in the antibody producing cell, minigene number 1 and minigene number 2 are now touching each other. They moved. And in fact, the fact that they moved is really the key thing here. So you can see that before they were apart, here they're together. There was some sort of movement in the DNA. Tonegawa took DNA from a cell that was a non-immune cell and an antibody-producing cell, and he isolated that DNA. The DNA was too long to run on a gel, so he cut it up with restriction enzymes. You don't care about that. You care. I'm mentioning it for 2 reasons. One, the band sizes, if you look at different versions of this experiment, sometimes are wonky, and the reason for that has to do with the restriction enzymes. So actually the band sizes don't matter to you. If it's big or small, doesn't matter because it has to do with where the restriction enzyme cuts. The other reason why this is interesting, this got him a Nobel Prize. When he did this experiment in 1976, there were only 2 restriction enzymes known. He just used one and picked one, and we now know that if he had used the other one, the experiment wouldn't have worked. So he basically took all the DNA, cut it up with restriction enzymes, and ran all of it on a gel. And then he took this mRNA, and he labeled it radioactively. So he made radiolabeled mRNA. Ideally, the radiolabeled mRNA should be the complement of the DNA that encodes it. Should be able to bind by complementary base pairing. And so he then put that radioactive piece on his gel. He allowed it to hybridize. Actually, it was on a membrane because gels are flimsy, as we talked about last time. And what he noticed was that if he looked at a part of the mRNA that detected the constant region, he got a band. He got a band in both cases. But if he looked at a piece of mRNA that bound the whole thing, in the non-immune cell, it bound in 2 places. It bound to 2 pieces of DNA, which now we know might look like this, where half of it binds in each place. And I don't have enough room to draw things. Here the front half's binding, here the back half's binding. I just don't have room to draw it. So it actually binds in 2 places, so you get 2 bands on the gel. And in these cells, it binds in 1 place. and you get one band on the gel. In fact, the product DNA has changed. The DNA that binds to this transcript has changed between these two cell types. This was used to prove the Minigene hypothesis. There is one other sort of prediction of the Minigene hypothesis. The other prediction of the Minigene hypothesis is that this DNA in between is thrown out by the cell, and in theory, maybe you could find it. And so he looked at these antibody-producing cells by electron microscopy, and he found excess loops of DNA that had been discarded. And so, in fact, he could even find the thrown-away DNA that had been discarded in these cells. Tonegawa, as I said, won the Nobel Prize for this work. Tonegawa is still an active researcher. He's currently at MIT, though he wasn't there when he did this. And after doing all of this work, Tonegawa basically decided that immunology was over and all of the interesting questions in immunology were solved, and he started doing neuroscience. And so he is now a very active neuroscientist. Yes?
With the random question, but how does, for lack of a better term, the cell know where the different segments are and how to bring them together?
That is a great question. I am going to make you ask me that question again on Monday, because that's a specific thing we're going to talk about on Monday when we go into the details. But Emilio's question is, how does the cell know where the Minigene segments are to cut and paste and combine them? Right? What do you think, what is implied in the answer? What do you think my answer has to have as a part of it? Yeah, Malik.
There's a unique sequence,
Yeah, there's got to be some unique sequence or some way that they vary from other things. There's got to be a sequence that marks them. And that sequence that marks them is one of the things on the list for Monday. So this is awesome. Awesome questions. I'm very excited. So as I said, the way that this is going to work with our Minigene hypothesis is we've got many minigenes that are going to combine in each B cell in order to make our antibodies. So to answer some of your previous questions, this process of recombination, remember recombination is basically cutting and pasting DNA. So we are breaking the cell's DNA and pasting it back together here. Happens twice in our B cell, once for the heavy chain and once for the light chain. And then we combine different heavy chains and light chains and get a lot of options. The heavy chain contains a segment called a V segment, a segment called a D segment, and a segment called a J segment, as well as a constant region. The first one is mu, the next one will be delta, gamma, epsilon, alpha. So heavy chains have a V, a D, and a J that they put together before the constant region. And so in the end, we get a V, a D, and a J put together. Light chains only have a V and a J and a constant region. And so that, I hope, answers one of the questions that you guys were asking about where there's a D and when there's a D and what's up with D. D is found in heavy chains. It's not found in light chains. So you can see that each of these segments encodes different amino acids in the final protein. So for example, in the heavy chain, the V segment is encoding about amino acids 1 to 101. The D is about 102 to 106. The J is about 107 to 123. You can see that in the light chain, the V is encoding about 1 to 97. The J is about 98 to 110. And then all the rest, all of the constant region, all of the tail parts, are encoded by the C regions. Yeah, Molly?
This might be dumb, but does VDJ recombination always happen V then D then J?
It does not. It in fact first goes D to J and then goes V to DJ. So yeah, that's coming too, don't worry. But yeah, it is event first. We're going to do D to J, and then we're going to do V to the DJ when we get to that. Yes. This is like awesome. These are great questions. You guys are like listening and thinking. I'm so happy. So one of the things that sometimes people will ask is about, okay, so why do we have to do this rearrangement process? Why can't we just get like a mega transcript that has all the V's and all the D's and all the J's and all the, why can't we just get super transcript? And this figure from your textbook, I hope, shows you a little bit about why. This figure from your textbook has the actual lengths of DNA that these sit apart on the chromosome. They are all kind of next to each other. In B cells, there are two different kinds of light chains. One's called lambda, one's called kappa. Kappa's on chromosome 2, lambda's on chromosome 22. In both cases, they just have V's and J's. They're arranged a little bit differently. We don't care right now. Heavy chain is on chromosome 14. And what you can see is that these segments are very, very far from one another. So you can see that, in the kappa segment, between the V's and the J's, there are 23 kilobases of DNA. RNA polymerase is not going to go across all the V's and get to all the J's. That is just way too long for one transcript. And you can see there are similar lengths between all of this. The only way that we can have transcription going on here to make a protein is if we cut out some of that intervening DNA and put things close together so that we can actually get a transcript. Similarly, you can see that being true for the heavy chain. One thing to notice is that mu, which makes IgM, the IgM constant region, and delta, which makes the IgG constant region, are relatively close to D and J, whereas the other constant regions are so far away. And so that's going to be a whole other issue at some point, because that is a long distance. Yes, Marina?
Is it possible to do recombination between chromosomes, or is it?
We'll come back to that. Great question about recombination between chromosomes. And so this is just sort of one other thing showing you that. In fact, there are promoters in front of every V region, so in theory you could start transcription there. But you can't really get a transcript that's long enough. And those promoters interact with an enhancer that is downstream of the C region. So only after you cut the DNA and make it shorter do you actually get the promoter and enhancer to work together to allow for some transcription to happen. And so then you get the RNA of just the V's and J's that are combined together, and you never get the whole big long thing. So that's the other reason why this recombination is important, to get the promoters and enhancers together and to make it so you actually can do transcription. Yep, Nadia. So it actually basically just sits in the cell and eventually gets degraded. So there are you can detect it pretty easily by microscopy. T cells, spoiler alert, are going to do this whole process too. When I was in grad school, one of the things that our lab used to do was we had this way to do PCR to detect that leftover DNA to see if a T cell had just been made or if it had divided a bunch and had degraded that. And so, in fact, it's in a cell that's just undergone development. It's sitting there in the cytoplasm. Yeah, Molly. It could be a ton of DNA to throw out, yes. Potentially. So one thing that you may be noticing here, and that you will notice throughout our discussions of VDJ recombination, is that the immune system and efficiency do not go together. This is an exceptionally inefficient process. Yes, we are throwing out tons of DNA. We are doing this on a numbers game. You don't know which V and D and J you need. You just make one of everything and hope that you got the one you need later on. This is completely inefficient, but it gives you a broad immune system that is ready to go before pathogens come. So when those pathogens are trying to evolve and out-replicate us, we have this advantage. As I mentioned to you guys on the first day, having the ability to protect sort of kids from infectious disease was a very, very strong selective pressure. And so even if we are, wasting DNA, and as you'll see later, wasting cells, we are in fact, and so probably wasting ATPs, that's actually more evolutionarily favorable than having your kids die because the new virus evolved a thing that we don't have a receptor for. So we just make every receptor. Yes?
So you said it was a numbers game and that there's potentially one cell that can produce an antibody for any given antigen. So what stops this number game from making stuff that could lead to autoimmune stuff?
Things that are self-reactive? Nothing. In fact, those are all made too. And so if I'll just go quickly. This is, again, a thing that we're going to talk about later, but I love that you guys are coming up with these questions. So I've shown you guys this figure a bunch of times. In fact, we make that huge number of diverse antibody-producing cells, including all the self-reactive ones. And I put a little white box over the label at the top of one step of this because I wasn't going to tell you about it. But in fact, there's then a deletion step where all the cells get checked and we purge the self-reactive ones. And so there's a deletion step here. So you can see number 1 and number 4 didn't keep making it. We delete all the self-reactive ones, and then we actually release them from primary lymphoid organs to be able to look for antigens. So again, moral of the story, immune system, efficiency do not go together because we just make every single possible antibody and delete out self-reactive ones. Great question. All right, so as I said, the recombination is going to allow for transcription to happen. And so in the end, what we will see is that to make a heavy chain, we will do this recombination process. First, we'll recombine D with J. Then we'll recombine V with DJ and get this shorter piece of DNA than we had before. Then we'll transcribe that mRNA. We'll splice that mRNA to bring the constant region parts closer. And then we'll translate to make a protein with the heavy chain that has been encoded by these parts. We will similarly do this for the light chain. With the light chain, we're going to recombine a V and a J. We'll then transcribe that product, splice it to put the constant region close, and then translate to get a light chain. And then we can combine diverse heavy chains and light chains in order to get our overall diversity of antibodies. If we look at the math of this overall, we have about 45 V segments for heavy chain. We have about 23 Ds and about 6 Js. If you multiply those together to get all the combinations, that's 6,210 heavy chains. And then we've got kappa or lambda light chains. We can get 205 different kappas and 165 different lambdas. So if we multiply the heavy chain number by the light chain number, combinatorial diversity gives us 2.3 times 10 to the 6th different antibodies off of, what is this, less than 6,500 mini-genes. So it's 6210 plus 205 plus 165 pieces of DNA gets us over 2 million antibodies. So this combinatorial diversity is really working out for us in terms of saving on genes, getting a big diversity. I told you before about the complementarity determining region section of the antibody. So I told you that all of our antibodies are made of these immunoglobulin domains with the 7 antiparallel beta sheets. And that the variable region of our antibody isn't like similarly variable all the way across. Some parts are pretty conserved. Some parts are really, really variable. We call them the hypervariable regions, or HVRs. Those hypervariable regions bind to the antigen, like 3 little fingers. They are the loops that come off the beta sheets. And so you can see the heavy chain and the light chain come together with these 3 loops to touch the antigen. Those are the complementarity determining regions. Those are the parts that are super variable. So where are they in this whole process? Well, the complementarity determining region 1, CDR1, otherwise known as HVR1, is just a part of which V you pick. And number 2 is also just a part of which V you pick. But CDR3 is actually encoded by the junction between the V, D, and J, or V and J you pick. And so CDR3, in fact, has by far the most diversity because it is actually made by the join of those V, D, and J. So every antibody that picks V number 73 has the same CDR1 and CDR2, but they'll all have different CDR3s based on which D and J they've combined with. So we got a pretty good amount of diversity here for our antibodies. But there's another piece to diversity of our antibodies. Because we got to 2 times 10 to the 6th, but we didn't get to that 10 to the 16th number yet. And so there's one other part of this. And that part is junctional diversity. Junctional diversity is basically a fancy way of saying that when we put our 2 mini-genes together, when we put our V and our D or our V and our J or whoever together, in this case it's V kappa 21 and J kappa 1, V kappa 21 being pink and J kappa 1 being yellow. When we put them together, it's not precise. We can add and subtract base pairs. And so in fact, if 4 different developing B cells all picked V kappa 21 and J kappa 1, they could still end up with 4 different antibody specificities because they might add or subtract different numbers of base pairs when they do that joining. So when we actually make the junction, when we join all of our gene segments, we have this additional process of junctional diversity that's going to occur. And so when you add that junctional diversity on top of the other combinations that we saw before, that's how we get to those super huge numbers of different antibody specificities that are made. All right, so there are a few other pieces that I want to point out here. So one of them I've kind of already touched on. As I said, each B cell, when it develops, recombines a V, D, and a J segment. And this process happens in the absence of antigen. And it happens all the time. So again, next time your mom yells at you and says you're not doing anything, you can be like, hello, Mom, I'm doing VDJ recombination. Because, in fact, you are. In making new B cells. And you are doing that all the time. Your cells are basically randomly picking V's, D's, and J's on their heavy chain and V's and J's on their light chain to make new combinations, to make new cells. You're just making them all the time. You will test them so that only the ones that are not self-reactive get released, and then they just go around your body looking for their antigen. And if they find their antigen, then they will divide and expand and make more of themselves. If they don't, if they are like number 3, one of my immunology colleagues will say, you know, they will go around forever with unrequited love. Just going around and around and around, and they will never find their antigen if you never get infected with that particular microbe. And so this is sort of what that looks like again. At the beginning, you have one cell that's making your antibody of interest. When we see antigen, that cell divides and makes way more of itself. That's how we get that amplified response. The response will contract, but you will have more cells than you did at the beginning. And that's one of the reasons, though there are many, that the response is able to be so much faster the second time around, is because you have more cells. But notice at the beginning you had like 1 cell. You didn't have 0. So your immune system does not make antibodies in response, does not do VDJ in response to infection. You did the VDJ before. It's just that you expand that cell afterwards. This is why immunologists stopped using the terms acquired immunity, because you didn't acquire anything. You had the cell way beforehand. That cell just adapted, which is why we go with that term. So knowing that this happens in the absence of antigen is really, really key. This process of VDJ recombination is one of the things we use to define adaptive immunity. So whenever you see these charts of what's the difference between innate and adaptive immunity, you often see with adaptive immunity, there's something about diversity, a highly diverse repertoire, a very large number of receptors arising from genetic recombination. And then for innate, it always says conserved germline encoded receptors, which means they're just normal things encoded by normal genes. They don't do VDJ recombination. One of the things that's part of the definition of adaptive immunity is this VDJ recombination process. This VDJ recombination process is actually a hallmark of all jawed vertebrates. So it looks as though, in fact, there was about 500 million years ago, 550 million years ago, the ability to do this was gained by gaining the enzymes that will do this process. And so all jawed vertebrates do VDJ recombination and have this adaptive immune process. Sometimes this event is referred to as the immunological Big Bang, evolutionarily. There are some people who can't do VDJ recombination. These people have diseases usually known as SCID. There are a few different types of SCID, or severe combined immunodeficiency. These people have no B cells, no T cells. They basically have no adaptive immune system, though they still have an innate immune system. And thus, they're able to live in a very, very controlled environment. They are extremely susceptible to infections, but if we control their environment, then they're going to be okay, and there are some treatments that we have for these people. Oh, sorry, Alina. This is boy in the bubble, yeah. One of the really famous genes for this is on the X chromosome, and it's X-linked, so you see it in boys pretty often. So there are a couple problems that I want to point out. One of these is a problem in terms of innate versus adaptive immunity, just to point out why adaptive immunity is so awesome, and why innate immunity is so awesome as well. They're both awesome. Everything's awesome. With antibodies, they are extremely specific for their epitope. They have the exquisite specificity, as I have mentioned before. But what that means is that our microbes are going to be able to mutate pretty quickly to avoid that exquisite specificity. And so here you can see our virus might be able to mutate its purple membrane protein to avoid purple antibodies, and on and on. And so while this specificity is amazing, it also is a little bit of a downfall of adaptive immunity. And that's why it's nice that we have innate immunity, where we see binding to conserved things that are not easily alterable by mutation. And so even if microbes are sort of messing with us and trying to get around the adaptive immune response, they are not totally able to get around the innate immune response. So we had some questions earlier. Marina asked about, can this go wrong? Can we get recombination in the wrong place? If you were to make a list of all the tissues of your body and how frequently there are cancers of those tissues, number one would be skin. Skin cancer is really frequent. And if you think about it, that makes sense, because skin's in contact with the outside world. It's going to get mutated really easily, right? And if you look down that list, you'll find a whole bunch of tissues that are in contact with the outside world and the immune system. And the reason why we see so many different types of leukemias and lymphomas are generally because we see VDJ recombination errors. And so in fact, cells at different stages of this process, this is showing you different types of B cells who undergo recombination errors, can end up becoming the progenitors of a number of different tumor types. So if this goes wrong, you end up with cancer pretty easily. In fact, there are some very famous situations where we can have VDJ recombination of the immunoglobulin heavy chain happening on chromosome 14. And if we break that DNA and it invades and tries to attach itself to the wrong other chromosome instead of to the chromosome it's supposed to, so yellow is supposed to attach back to yellow, but if it decides to attach to blue instead, sometimes we'll get this chromosomal translocation where a very famous gene called MYC will actually get put under the control of the Ig heavy chain. This leads to something called Burkitt's lymphoma. There is a similar translocation that pretty frequently happens with the light chain that leads to a chromosome that's actually very easily visible under the microscope called the Philadelphia chromosome. And all of these are VDJ recombinations. So yes, Marina, there can be situations where the joining happens improperly. And what I hope that you realize from this is that this is a dangerous, dangerous process. As a result, this process has to have a whole lot of regulation. It needs to have the special sequences or signals that Emilio mentioned so we know where to break the DNA. They're not just breaking the DNA any old place. We need to have special signals to join the DNA. We actually need to have holding proteins that let it hold it close to each other and don't let it go any old place it wants. There are going to be tons of regulatory steps. And those regulatory steps and all of the details of this are what we're doing on Monday. Is we're going to go through the nitty-gritty details of all of this process on Monday. Your textbook has this table of some of the proteins that are involved in this process. Some of them I'm going to talk about a lot. Some of them I'm just going to do as a drive-by. So RAG 1 and 2, we're going to talk about quite a bit. They're going to be the stars of the show. TDT, we're going to talk about quite a bit. Artemis, we're going to talk about quite a bit. RAG and TDT are only found in B cells and T cells. Everything else on here is actually found in all cell types as part of general DNA repair. We just use it for VDJ. So the other ones I'll sort of mention briefly, but RAG, TDT, and Artemis are really going to be our stars of the show. And so this is why Monday is going to be a lot of detail of a lot of things. And this is why you need this many details, is because if this process gets messed up, it goes really, really, really bad. Yes, Kay?
Is this part of why people who already have chromosomal disorders, like I know people with Down syndrome have a higher rate of getting leukemia?
It can be part of what's going on there. There are a few things going on there, but that can be part of it, yes. And this is an overview of the mechanisms that make B cell diversity. This is table 6-3 from your textbook. And it's a cropped version of table 6-3 from your textbook. Because this table actually shows you all the mechanisms of B cell diversity and T cell diversity and compares and contrasts. Obviously, we are not ready for that yet, but it is a nice summary that we're going to see heavy chains, we're going to see light chains, we're going to see VDJ, we're going to see junctional diversity, and all of those types of processes. Some of these things that are on this list are going to differ for T cells. So on Monday, we are talking about B cells specifically. It is going to be a lot of molecular detail on Monday. I am going to be drawing DNA sequence up on the board a bunch on Monday. So make sure that you are ready for that. You might want to go through this section of the textbook once over the weekend to familiarize yourself with it before you see it on Monday. So have a great weekend. I'll see you guys on Monday.